Hello, everybody, and welcome to today's class on learning R functions. We've already run R functions, which are called base R functions, and we'll see more of those today as well. But you can also write your own function. So if you find yourself doing a task over and over and over again, you can simply write a set of base R functions and their parameters in a row and for loops that we're going to go through and build your own function. So it could be a lot of code that you would have to write every time you wanted to do that task. And what's nice in R is you can bundle that into a function. And when we looked at the question mark of like mutate or other base R functions, you get a nice panel of what's going on here. And that's because somebody else developed a function for you. And you can build your own function and have it and give it to somebody else. So that's what we're going to focus on today. And the inquiry-based lesson here is going to be, we're going to learn how to write functions in R to intersect peaks between replicate chip files. So we've already run the NF core Chip-seq pipeline, and we have two replicate files, and we want to merge and say, what are the peaks that are common in both of those files? And that's something we're going to do a lot. So we would write a function for that so that we could just call up one name like you do mutate or any base R function. You can call up the name of your function and it'll run what you need to do. And so we're going to see what all that is today. And we're going to need the tidyverse and genomic ranges and intersect functions loaded. So I'm just going to run those and get that set up. And so, you know, why would we write a function in R and what is a function in R? And so here's a simple example right here where it says, okay, this function has two parameters and we'll have it, it's basically going to multiply two numbers. And so we give it a little bit of a description, and then what it's going to do is set a object fun for this function that we're going to run, which is, and the R function you'll see turns red here because R knows that it's creating a function, and that function is going to need two parameters, X and Y. And then what the function is going to do is going to happen between these curly braces. This is just almost a cartoon example. You'll see a full example down here in a second. Well, this is just to say you can name a function. So we can name a function intersect peaks is what we're going to go over today. And that function is going to have two parameters that need to be inputted, X and Y, and then we'll carry out a function in between there. And math is a great example of this because essentially you map inputs to outputs in math. And so that's exactly what a function is going to do, is say, what are your inputs? It's going to do a function on them, and then it's going to produce the output. So this function is pretty simple. We have fun telling R it's a function with two parameters. And then now we're going to set another object here, answer, or ANS, which is just X times Y, and return answer. So now we can just type fun, and all of this will occur. All this between the brackets will occur here. Just simply X times Y for this. And then we're going to get into a much more complex one below. And then, so what's nice about this is now you can just, R now knows what this is. So let's run this function. Okay, so now we can simply put two numbers in there, and we can see that it returned 8, that 2 times 4 is 8. We can try a negative number, and let's get the same thing, we'll get -8. So that's this very, very simple function. And the key thing is to know the action is happening between the curly brackets, and you can have other functions inside of these functions, and that's what we're going to see below. But that's just one nice, simple little example. And when you mark these down, as you've seen in the help viewer pane here, where you get object summaries, it's really just, there's a common annotation here that you use where you give a very sort of generic high level explanation of the function. So this is a function to multiply two numbers. At description is also recognized by R, so we can do this here. And then this function will multiply the values of X and Y. So what are the parameters? And that's always what you find in here. What are the things it's looking for? So it's looking for an object, and it's going to do this to that object. So we need two parameters, x and y. And so then you give an example of what the code is below. And you can see that here in our util file. This is where you would keep these, why we made this util folder with intersect functions, for example, is where the functions we're going to go over now are. And so now R knows where they are, and I can just type in intersect peaks and it runs a bunch of code. Just like if I run fun, I get to multiply two numbers. Okay, so this is some annotation. You can go through the markdown here. I'm going to go through this just on a sort of a high level. This is a sort of going to be a long, it's a function that's a large set of code. And so it's going to take a little bit of time to go through it. I'm going to go just over the sort of high level concepts. There is a bunch of markdown here to go through what each step is doing and what's happening. So feel free to take your time with this. I'm going to go through it, not too fast, but just sort of focus on what the high level, the points are. Okay, so like we made a function before, we're going to make a function create consensus peaks. And what consensus peaks will just simply mean is that if there was two peaks that overlapped in the same part of the genome in two different replicate chip experiments, we're going to take only the peaks that show up in both replicates so that we're a little more confident that they're reliable peaks. And so we would do that quite often. And so now in the future, after we go through this lesson, we can just write create consensus peaks and it'll run that function. Okay, so let's get in here. So first thing we're going to do is the only parameter to this function right here is the file path to the peak files that we got from Chip. So we just outputted those 2 or 4, however many you wanted to run, peak files, the .broad peak files, and they're here for our class. You're going to have to set where this file path is. That's why it's the only parameter in this function. So by the end of this class, you'll be able to intersect peaks for as many files as you want, as long as you set the parameter of the file path of where those peak files are. Okay. So that's where our peak files are. So let's actually set that and run just that. And we'll see in here we have broad peaks files here. And if we look at it a little closer, we should see that it's in the right spot. So it gave us the right value for that. Okay, great. So now we, a while back, went over list files, which is simply a base R function that wants to know where the files are. And this is just a parameter to say that include the full names. And this FL object we're making here is simply just going to be a list of files. And that's sort of it gets easier as you go. But first, what we're going to do is set a set of objects that we're going to call in for loops and other things. So the complicated part's going to come below, but we want to set some of these objects to have those for loops course with the code. Okay, so we're going to have a file list variable or object, and it's just going to be essentially all of the files that you have in your peak folder. In this case, and it's always good to have this environment open to look here, is that we got 1064. That's exactly the number we're hoping for. That's how many peak files that was called by Max in our next flow run for the full experimental design of this class. And so now we've started this function. And so it's important to remember we're inside of a function because of this curly brace here, but we can also have functions run inside of those functions. And that's what we're going to start to see here. In fact, you can have a function run, complete itself, and then still carry on what's going on in here. So really, once you fully close this curly brace, then you're going to stop this function, which is going to be a bit down, a bit of code down from here. So we have this FL, it's our 1064 where those peak files are. And so now what we're going to do is have what is called an unnamed or sort of generic function. So you can just make a function on the fly in R as well. So we're just gonna make a function X here. We're gonna call it TF name, because or DBP name, it would probably be better nomenclature at this point, but TF and DBP we're gonna use interchangeably. So let's say TF name, now we get to see a base R function called sapply. And what sapply's parameters are, and you can go through it down here a little more, is what string lines or what character lines do you want to apply a function to? And this is pretty powerful. So we can say, oh, these 1064 files here, we're going to apply this function X. And then we're going to tell it what function X is. And so sapply then will go through all of these file names and perform function X. So you can probably guess what we're trying to do here is take information from the file and make a new object. And that's exactly what we're doing, is to say we want to know what the DNA binding protein DBP TF is for a given file. So we would do that here. And now we're going to see a new piece, a new base R function, string extract. And what string extract is, and don't get too worried about all this, there's markdown here where you can actually put it in a website and it'll tell you what this is doing. But this is regular expression that we saw a little bit of before. And string extract requires the function, the object you want to run it on. And right now that object is function X. And it's X in X. So wherever, as it's going through each of these 1064 files, it's going to look at entry X, and it's going to use this regular expression. We're not going to go into this for the purposes of this class, but it's basically saying match one of these guys with the exact case and get dot broad peak, so that we're going to end up with that file name simplified down to just sort of the DNA binding protein's name in the file. So it stays attached to the file. And then we're going to use string extract again on the object we just created, file name, and use some different regular expression to make all the DNA binding proteins that all the names of the DNA binding proteins associated with the file. So let's run that here. So we are running the function TF name, and it's going to be an sapply and string extract combo. Again, I'm going over this kind of fast and high level, but it is marked down sort of what I've been saying. Okay, cool. So we got TF name here. And as we'd expect, we have 1064 files, and we have TF name. And what we're doing is we're building sets of information that we can use in for loops and nested for loops down below. We're just establishing kind of the information we want. We want to know where all the files are, and we want to know the name of the DBP for those files. And so we now have that information. Again, in here, you can paste in this regular expression code here, and this website will tell you exactly what it's saying in more user-friendly terms. Okay, so now you can see that we have replicate files for each chip experiment. And so we want to do a R function called unique. And what unique is going to do is say, okay, instead of 1064, it's going to collapse these two into one. And so you'd think this would shrink in half. It's not going to exactly, but don't worry about that. So it's going to say, how many unique names do you have in TF name? And that's why we sort of started to build these things up, is so that we can start to use them, and then we might want to use them in the for loops to index. So imagine we could have unique TF and then have that associate with both of the replicate files. And so we can call that DBP, and then it'll know that there's two files associated with it, and then we can have an intersect go on there. So we'll run unique TF. So we have not only all the names of the TFs in the files, but now we're just going to have the number. And so you can see that shrunk quite a bit to 486. That's because there were some that had like 8, 10 multiple replicates. Okay, so now here's where the function's really going to start And I'm going to go through this line by line, but it's not going to run because it's essentially a function that we're writing. And so all of this, what we just made here, are going to be inputs into the function, but we are still in this initial curly brace, which is establishing what this function intersect peaks is. Okay, so we are going to make what we're going to call consensus peaks is the merge of those two replicate files, or however many replicates. The peak has to be in all of them. Okay, so we're going to establish this, but it's going to be left empty, and it's going to be populated by this for loop below. But we'll sort of make it here just for one more level of simplicity before we have to run all of this. Okay, so what we can see now is we have consensus peak list. There's nothing in it because we haven't run the for loop. And so let's go through this line by line. Again, we'll have it marked down for you here. So we're starting a for loop, and we're saying for I in 1 through length unique TF. So this is saying there's, in unique TF, there's 486 entries of unique DNA binding proteins. And now we're going to do something. So for in all the things in unique TF, do this. And we're going to set the DBP to the index I. And so this is what I was mentioning before, is that index I is going to be one name, like ADNP here in this one, the first entry, but it's going to have two files associated with that name. So this is just the first index of that DBP here. And cat or C, we could paste 0. This is just simply in the terminal, we'll let you know which actual file is being run in unique TF. It'll say, now I'm running DBP. So it'll say, now I'm running ADNP, and this is just creating a new line after that for simplicity. And you'll see that run in a minute here. Okay, so now we need to make TF files, and that is going to allow us to know where all the files are. So we'll have this, the name of the DBP come up, and then all the files associated with it, because what we're going to do here is FL, remember, is the list of all the files. So FL has 1064. And we talked about this a little bit, but now what we're seeing something cool is we can index into FL with these brackets, but we can run a function inside that indexing. I don't think we've seen that yet. So we're just going to grep the DBP name, the unique DBP. So we don't grab two of them, we just grab one. And then we're going to grab all the files associated with that in FL, because there will be multiple entries for ADNP1 in FL. So again, you can go over that again, but it's basically we're going to make TF files so that we know what files, multiple files, are associated with each DBP here. Okay, and now in order to merge peaks, we have to have at least two files. So we're just going to add an if statement here. And remember, we started this for loop here. I know this gets kind of bulky, but we're right here now, and we're going to start another for loop, another part of this function in here. And this starts to get hard to keep track of where you are with these guys, but I'm going to point out a little bit just to say that we started populating consensus list here with this for loop, doing everything between here, but you notice this guy's in the same direction. So we're going to have to wait for a few closing commands before this stops. Okay, and so back to, if you have to have at least two files in order to intersect two files. So what it's going to say is, if the length in all of the files, the TF files, the 1064 we produced, if it's greater than 1, you can proceed. If not, it'll just stop, or just will ignore it, because we're not adding any other commands to it. So what now is going to happen is, if this is true for the file that, or for the unique, for the DBP, that it has more than one file to intersect, and then we'll make peak list again is an empty variable here that's going to be populated by a second for loop. Okay, so this is getting where it gets tricky, and why I was pointing this out up here is that we already started a for loop for I. And now what you can do is in that same, so we did that, and now we're doing something below that in here, and now we're gonna do something else in here. And so what's happening is we're running what's called a nested for loop. So we're still running the first for loop for I, and now we're gonna do a second one, and we have to set a second variable, and we'll use J here. Okay, so for J in 1 through length of all the files again, so a very similar start to the first for loop, but using J now to index to do what we want between these two curly braces. So I hope you can start to see how when you build a function, it's really kind of like building a tower of multiple base R functions into an actual function that you're going to use and call. Okay, so now to make that even a little deeper is that we're calling functions inside of functions in nested for loop here. And now what we're going to do is make an item called peak list, an object peak list. This is where we're going to start to get into where our peaks are inside the peak file. So right now we're reading the file names and that kind of things, but we have to get inside of them eventually. And so we're going to start that process here, where we just make a peak list where we run a function read peaks. This is a function we've already made, just like we're making this function here. We're now making a mini function inside of the function we're making, but we already know, R already knows this function because we sourced it up here in our util intersect functions R. So it already knows what that function is. And that's commented down below here where you can, or commented out so you can go see what read peaks actually is. It's already in there, and you can see we do the same thing that we've been doing down this list. Is we're setting in read peaks as a function that does what's in between here. And it tells the parameters, and it's returning ranges. And we have we have an accompanying video on what I ranges are, G ranges. For the purposes of this video, I think it's important just to think of that when we look at these peaks and we want to intersect them, we're gonna have to know their coordinates. We're gonna have to know like where they sit, and that's a range. So coordinates you can think of as a range, but a range is interesting because it's not only the start and stop of where those coordinates are, but there's packages called genomic ranges that will also know what chromosome it's on. And that's important because if we had two coordinate intervals from where the start and the stop is, then if it was on the different chromosomes that had the same numbers, that would just get too confusing. So that's really the main point of ranges, as we'll leave it for now. Please see the other video associated with this one, that it's a range and a chromosome association with that range of where the peak is. So then we can use those we can use ranges in one file and another file and then cross those to make an intersection. This is going to look complicated and we'll go through it in the other video. But for now, we want to just sort of reorient that what we're doing is we've started a second for loop here, and we're making a peak list, which is now going to be in genomic ranges because we use this function read peaks that we've already made, and it's going to go into each of those file paths, and it's going to grab out where the peaks are and then name it by the DNA binding protein. So it'll be like peak 1 through 30,000 if there was 30,000 peaks. So each peak will have a unique name and a range of where it is in the human genome. Okay, so that's what read peaks is doing. So you can see we've called another for loop inside of this function, and now we're calling a function we've already made inside of this function. Read peaks will actually clean up to canonical chromosomes. We talked about this before. It's something to always keep in mind, is that there's contigs of the human genome that don't have formats that most programs like. They want the chromosome number, like 1 through 22, chromosome M, X, and Y. There's a bunch of other things in there. And so read peaks will already clean that up for you, but just to sort of, for posterity, we'll do it again. It's a bit redundant. Okay, so now we're still in here in our nested for loop, and then we're gonna do another for loop inside of that nested for loop. And so this looks really complicated, but just think of it as an intersection. It's not that bad, and it's marked down here. You'll see a couple new functions. So again, we're doing for I and length through peak lists. So that's just really going to be 1064 things, but it's going to have the ranges associated with it. And so we can go into peak list I, because that's where we are, and the initial for loop, and say peak list I, where which, and then seq names is really handy because it's going to find the names that are in canonical chromosomes. So you can think of it as kind of like grep. We probably could have done a grep thing here, but it's saying seq names in peak list I that are in canonical chromosomes are the only ones that are going to pass into this index. So it's a bit much, but it's not too bad. Check out which and seq names, and then this should make more sense. And this is just so handy in our formatting. It's very natural language to say, tell me which of the peaks in the peak list only have the names of canonical chromosomes, and we set that up here with just a list. So this is going to go check back up here and say, okay, did you match any of these? If so, then I'm going to keep you in there, and it will essentially get rid of all of those contig peaks. Okay, so we're getting close. So we're now into the final peak set. So we're moving to make a new object in this nested for loop that is going to intersect peaks. And this is the function we're making right now. So it's going to run itself essentially because we've sourced it. But this is, in the end, the function that we are making right now is intersect peaks. An intersects peak parameter, all it needs is a list of peaks in which to intersect. And so where we kind of are now in these nested for loops is we've made a set of a for loop to go in for each unique DNA binding protein to find all of its associated peak lists, and then grab those so it knows in that one and put it into a second for loop, which is where we're about to end the second for loop, where we use intersect peaks on that peak list we just generated above. Okay, and we want to make sure that our peaks have peaks. So we're just making an if statement similar as we did above, that you had to have two files in order to intersect. And again, this is all sort of marked down here. Intersect peaks is shown here. And this is again going to be more familiar in the ranges that when we read peaks, we moved them into genomic ranges space, or ranges that knew where the chromosome and where the peak start and stop was, as long as other features. And this is saying to take the union of those two things, essentially. But it's all written out here. I'm going through this sort of quick on a high level. Please pause and read all this to understand more what's going on in intersect peaks. But it's essentially the function we're making. Okay, so now we're going to say if the length of the peak sex is greater than 0, so if it's less than 0, it's just going to ignore it. It's only going to move forward. Again, here's how we know where things are starting to happen, is the open curly bracket. And then we're going to make a column. We did this in the first R lecture, where you can just make a new column by using this index cache sign here. And so we're going to make a new column, and we're going to paste in the name of the DNA binding protein that was in this for loop, this first of 1064 iterations, or sorry, first of 486, that it's going to take the DBP that it's currently investigating and underscore, and add an underscore to that name, 1 through the length of final peak set, which is what we just created here, which is the intersected peak list, and it's our final peak set of merged peaks. So what it's going to do is just essentially add the name of the DNA binding protein to each of the peak sets. that were just created. And then this will list those, list is the base R command to look up, but it's really just going to list everything that's in final peak set up here, and add the names. So we still haven't finished, or we've finished this by adding the names to them. So we're going to run the names function on consensus peaks through the length of it, and we're going to add the DNA binding protein's name. I know that sounds a little around the elbow. It probably is. But what we're essentially just doing is saying, here's our consensus peaks. We want that thing to be populated with the final peak set, and the names associated with that are all of the things in the length of consensus peaks, and adding DBP, which is the unique name here. And so then in the end of a function, we need a return. That was a long run through many loops and calls of functions inside of functions. But essentially, what we're going to return is consensus peaks, which we just created here in the final list. So then after we return that, we're going to have, well, let's run it and see. I'll show you. So again, we made all of these here. We made consensus peaks, and that's where we just started from, is we're going to populate this consensus peaks, and it's going to turn into final peaks, and then consensus peaks again at the bottom to finish it off. So if we run all of this here, oops, sorry. We can see why we sort of added that paste here, just to say, it's kind of a nice little debugging aspect to say, oh, okay, if something doesn't work, it's going to stop running and error out. And you can just see where it is in the list, might help debug a little bit. Also, it might just be annoying, and you can just delete it if you don't want to know where the computer is currently running. And so what we're doing here is we're creating all of these, but we haven't returned it into a list here of consensus peaks. This is not needed. And so as soon as this is done running, which is probably going to take a minute, and we don't want to do this for the whole video, is I'll just leave you with, we would run this last little bit here to return consensus peaks, and consensus peaks will be a long list here. And so now consensus peaks is going to be an object that is going to have the consensus peaks for all 486 of these things. So it's going to be quite a big file. So what we're going to do is print each one of those consensus peaks or merged peaks files out into a folder called, we already have results in our class one. If you don't, you're going to want to make a results folder in your working directory, and then make a new folder called consensus peaks, which is what we just created. And then again, just a simple for loop where we know where this starts and stops is what's happening. And so again, through the length of the consensus peaks, that should be consensus peaks. Apologize for that. That for each of the I in length of the files we just made that we're making right now, we're going to invoke our track layer, which is a genomic ranges function in the associated video that we went over. I suggest watching that first before this video, but our track layer is going to know that you have a unique DNA binding protein name, you have a merged consensus file, and it can export those coordinates into a nice .bed file so that we can add that for analysis. And just a refresher, when we went over this in the UCSC lecture, but that a .bed file is simply just at least chromosome start and stop of where a peak is. And now each of our peaks within each file has, like, let's say there was 10,000 peaks that merged. There's going to be 10,000 entries where each peak is going to have a unique start and stop and chromosome position. Okay, and that's also just simply our track layer export. And you can go through this while I'm going to leave this running so we don't have to wait for it. But essentially all of the for loops for each unique DNA binding protein are running right now. We can see them going. And then after that, we just simply run this bit to print them out into the results folder. So we have to tell it what to export first, and that's the consensus peak it's on. So for instance, right now, H3K36, this one, would be being pasted. This information will be pasted with the bed.bed to it in this folder. So essentially just creating a file path and where to put the exported file. Okay, so there you've created your own function, and now you can see that in our util folder here, we have intersect functions, and we can find intersect peaks right here, where this is what was happening in G ranges, where it was simply combining the peaks to find the unique overlaps within a given, between the two files or multiple files. So with that, I think we'll wrap it up. This is difficult. It will get easier as you learn each of these base R functions. Take the time to check them out in the help pane as you go along. This one will take probably a bit of time, but it is written down to what's happening, and as you can see, it's running. So as long as your file path is correct to where your peak files are for your chip, you now will have an intersected set of peaks. And you can see that why we would want to go through all of this work to name something intersect peaks, because it took all of this to get to do that. So now in the future, we're just going to say intersect peaks, and our util folder is going to know where that function is.